Hi, everyone. Today, we will dive into a new research paper that we just published regarding the differential roles of IL-4 receptor alpha and IL-13 receptor alpha 1 in eosinophilic gastritis. Eosinophilic gastritis, or EOG in short, is a relatively rare gastrointestinal disorder that causes chronic pain, causes nausea, vomiting, and dyspepsia, and is closely linked to allergies and elevated type 2 cytokines such as IL-4 and IL-13. Histopathologically, EOG is characterized by accumulation of mast cells and eosinophils, as well as thickening of the epithelial and mucosal layers, and if the disease is chronic and untreated, it could progress and become fibrotic. A big challenge in EOG is that we haven't fully understood the molecular events that drive this disease. To solve this, we developed a robust experimental model. The model is initiated by skin sensitization and challenge of mice with oxazolone, followed by intragastric challenges. Subsequently, we dissect the mice and put the tissues in different orientations to study the histopathology and tissue architecture. As you can see, the model is characterized by increased infiltration of eosinophils. In addition, we can observe elevated levels of mast cells, mucosal and submucosal thickening, as well as fibrosis. Thus, the model successfully mirrors what we can see in human patients. When we look into the transcriptome profile of EOG, we found a distinct molecular signature with upregulation of genes representing a mixed response of type 1 and type 2 immune responses, as well as epithelial activation and remodeling, while suppressing normal gastric functions like acid secretion and metabolic detoxification. Importantly, comparison of the transcriptome in experimental EOG and experimental EOE revealed that they have a distinct gene program, although both of them were induced by oxazolone. This suggests that we have disease and tissue-specific responses. IL-4 and IL-13 have key roles in multiple type 2 immune responses and are therefore key targets for therapy. Therefore, we were interested to determine whether IL-4 receptor alpha or IL-13 receptor alpha 1 have unique or overlapping functions in EOG. We found that if you block IL-4 receptor alpha, you can actually stop epithelial thickening and the infiltration of mast cells and eosinophils. Interestingly, however, when we tested mice lacking the IL-13 receptor alpha 1, we could block epithelial proliferation and thickening, but the gastric mucosa of these mice was still flooded with mast cells and eosinophils. This tells us that these two receptors play very different roles in how the disease progresses. This research opens up new possibilities for how we can target and treat EOG in the future. I want to finish by really thanking all of the dedicated lab members in the Tel-Aviv University in my lab, especially Anish De Silva, who spearheaded this project and is the first author on this study. Obviously, I wish to thank also all of our funding agencies. And if you wish to read a bit more, just go to the paper. Thank you very much.